Hello everyone, welcome to the second video on this AQA Combined Science Biology Higher Tier June 2022 paper. In this video I'm going to go through questions 3 and 4. So let's move on to question number 3. Question number 3, amylase is an enzyme that digests starch. So remember, starch is broken down into glucose by amylase. Which organs in the human digestive system produces amylase? So liver, small intestine, large intestine. That's not correct. Salivary glands, yes. Stomach, no, because it is acidic. It produces hydrochloric acid. And the third one, salivary glands, pancreas, small intestine. That is the correct answer. And stomach again is not the correct answer. Next part of question 3.1. A student investigated the effect of pH on the activity of amylase. This is the method used. Prepare amylase solution at pH 5, which is slightly acidic. Mix the amylase solution with starch in a boiling tube. Remove a drop of amylase starch mixture every 30 seconds and test it for the presence of starch. Record the time when all the starch has been digested. Repeat steps 1 to 4 using amylase solution prepared at pH 6, then 7, and then at 8. I believe this practical is one of the core practicals or the required practicals in AQA biology. So 3.2, what was the independent variable in this investigation? So remember, the independent variable is what you change, right? What you change, and that would be the pH of the solution. So pH of solution. Now while we are looking at variables, remember there are two types of other variables: the dependent variable and control variable. Right? So in this practical, what would be the dependent variable? That would be the time it takes for the starch to be broken down by the amylase. The control variable would be the enzyme concentration. Let's look at the next part of the question. Question 3.3, describe how the student would know when all the starch has been digested. So remember, in the question, they are adding starch and mixing it with the amylase solution, right? And here they are asking, describe how the student would know when all the starch has been digested. So they are asking, in the third point, right? Remember here they're testing it for the presence of starch. How are they actually testing it for the presence of starch? Using iodine, right? So iodine changes to blue-black color with starch. So going back to the question, how would the student know when all the starch has been digested? The solution or mixture, which is the starch and the enzyme mixture, would not change color to blue black. If there was any starch present, then it would actually change to blue black. Once all the starch is digested, it won't change to blue black anymore. Question number 3.4. Figure 6 shows the students' results. So as you can see from the graph, on the y-axis is the time taken to digest all the starch in minutes, and on the x-axis is the different pH levels starting from 3 to 9. Remember, they only tested 5, 6, 7 and 8, just those values. The question is asking, what was the optimum pH for the amylase? The optimum pH for the amylase is when all the starch would have been digested with the smallest time taken. So that is at this time. So that is at 6.8 pH. Each of the boxes 0.1. So that's 10 boxes. So that's 6.8. Next question. A scientist did a different investigation. This is the method used. Prepare amylase solution at the optimum pH. So he's actually using a pH of 6.8. Mix the amylase solution with starch in a boiling tube. Measure the concentration of sugar every 10 seconds for 2 minutes. Figure 7 shows the scientist's results. So in the graph, you have the time on the y-axis between 0 and 120 seconds, and the concentration of sugar is increasing rapidly at the start, and the rate slowly decreases and then it becomes stable. So the question 3.5, how much time did it take for the amylase to digest all the starch? So at this point, when the line is actually just starting to become flat, is when the concentration of starch is actually remaining, the concentration of sugar is remaining the same. That's when all the starch has been broken down into the sugar. So time to digest all the starch, right? Again, this is 90, so each box is 2, so that would be 82 seconds. Next question. Determine the rate of sugar production per minute at 40 seconds. So how do you find the rate at 40 seconds per minute? So the answer has to be in arbitrary units per minute. So let's have a look at the graph again to find out what's happening at 40 seconds. So at 40 seconds, this is just below that 3.5. So you could take it as 3.5. So it's just below that. So it is actually 3.45. So you could, if you divided the concentration by the time to find the rate, that would only give you 1 mark. Remember, this is a 4-mark question. So in the question, determine the rate of sugar production per minute at 40 seconds. It's a 4-mark question. So simply finding the values at y and x and dividing it would actually give you those 4 marks. So there's a reason why it's 4 marks. Here you need to find the tangent. So let me draw the tangent and then show you. So I've drawn a tangent at 40 seconds, and the value, by drawing a triangle, the value of y is 2.4. So that's 4.4 and it's starting at 2. So I've taken away 2 from 4.4, which gives me 2.4, and the value of x equals 62. So let's work out the gradient. So right, gradient equals y over x, or the change in y over x. Remember, y was 2.4 divided by 62. This gives us a value of, so 2.4 divided by 62 gives me 6 over 155, which is 0.038. So I can round this off to 0.039. 0.039. So 0.039 is the rate per second. Now here they are asking for the rate per minute. So because you want to find it per minute, so you would multiply that value 0.039, multiply that by 60. So multiply this value by 60, it should give you 2.3 as the answer. So that would be 2.3 arbitrary units per minute. Question 3.7. Question 3.7. Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. It's a 6-mark question. So explain. So you need to give reasons how the structure of enzyme molecules. So remember what the structure of enzyme molecules are and how are they related to the effect of pH. So you need to say what happens to the enzyme structure when the pH is changing, and that how that in turn affects the activity of amylase. So looking back at the graph, right? Now remember, as the pH changed, so this is the very first graph that we are looking at, right? As the pH changed from 5 towards 6.8, the rate of reaction is actually high. So, and then 6.8 was the optimum, and after that the rate of reaction actually got slow, or it actually took much longer. So how do we relate all of this information into our answer? Going back to the question, explain how the structure of enzyme molecules. So for this question, you need to refer back and think about the lock and key mechanism as well. So let me write a model answer and then I'll discuss how I have answered the question. So here's my answer. I've written 6 points for the 6 marks. So going back to the question, explain how the structure of enzyme molecules is related to the effect of pH on the activity of enzymes. First point, enzymes are protein molecules with 3D shapes. They have specific shapes with regions called active site where a substrate can fit, and in brackets I've written down lock and key. I don't have to go into detail, I've mentioned it, which indicates the examiner that you know what you're talking about. So which are complementary to each other, meaning that they have opposite shapes. I'm referring to the active site and the substrate shape. Point 3, starch is a substrate of amylase. Point 4, at pH above or below optimum pH, which is 6.8 pH, the shape of active site changes. So substrate would no longer fit. At very high or very low pH values, the enzyme would be denatured. The last point, so at pH values other than the optimum pH, the rate of starch digestion by amylase decreases. Now the 6th point is very important because it addresses this last point. How is it going to affect the pH on the activity of the enzyme? So all of these points is actually slowly building up to what I'm actually saying in this very last question. If the pH value changes other than the optimum pH, then the amylase activity or the rate of reaction actually decreases. Before we move on to question number 4, let's have a look at the marking scheme. So question 3.1, the answer was salivary glands, pancreas and the small intestine, which we chose. 3.2 was pH was the correct answer. You could also say hydrogen ions. I would just simply stick with the pH. 3.3, iodine would not turn blue-black, or you could say the iodine solution or reagent would stay orange-brown. Remember, the original color of the iodine solution is this orange brown color. So what we said is basically this iodine solution or reagent would not turn blue black. Question 3.4, the optimum pH was 6.8 and the time it takes is 82 seconds, and here they are allowing you to have a range between 80 and 84 seconds. Question 3.6, tangent drawn at 40 seconds. So this is where you're getting the 4 marks, right? It has to be drawn at 40 seconds, and we worked it about 2.4. Here they are working it about 2.25. So calculation of rate of reaction based on this value, what they have worked out, their answer is 0.0375. We got 0.038, remember. Again, they have given a range, so your answer could be anywhere between this range. Now once you do that, you are trying to calculate the arbitrary rate of reaction in arbitrary units per minute, so they're multiplying it by 60 again, which brings it back to 2.25. So our answer was within that range of 2.1 to 2.5, right? Here this part is very important, right? If no other marks are awarded, allow 1 mark for simply dividing 3.5 divided by 40 and then multiplying it by 60, right? That would give you a rate of 5.25, which will only give you 1 mark. The 6-mark question, you're aiming for the level 3 answer. Here, relevant points, reasons, causes are identified, given in detail and logically linked to form a clear account. This is why I always encourage my students to write it in bullet points and number them, right? So that you can actually work out what happens after each of these steps. So what are they actually saying? Enzymes are protein molecules, so have 3D structures, right? Although they have given different bullet points, I have actually combined most of these bullet points, or in my answers I have actually got it in the points combined in sentences. Lock and key theory, have an active site, have specific shape. Shape of active site will only match to shape of substrate. Starch is substrate of amylase. All of this I have mentioned. At pH values above or below the optimum, the shape of active site is changed. Substrate can no longer fit into the active site. At extreme pH values, enzyme is denatured, so the shape of the active site is changed. So amylase can no longer digest the starch, and so the rate of digestion decreases. So that would give you 6 marks. So let's move back to question 4 now. Question number 4 is on photosynthesis. Photosynthesis is an important chemical reaction in plants. Why is light needed for photosynthesis? So remember, photosynthesis in terms of chemistry, it's an endothermic reaction. So photosynthesis is an endothermic reaction. So light transfers energy for it to take place, right? So all endothermic reactions absorb energy. Photosynthesis absorbs light energy. 4.2, what is the equation for photosynthesis? Right? So looking at the formulas, that's oxygen, this is water, that's glucose, and this is carbon dioxide, right? So during photosynthesis, a plant absorbs carbon dioxide, water to produce glucose and oxygen. So that would be the correct answer. Next question. 4.3, a student investigated the effect of different colors of light on the rate of photosynthesis at room temperature. So they're keeping the room temperature the same. So that is a control variable there. A student used pondweed in water. A piece of pondweed was placed in red light, then in blue light, and then in green light. So this is your independent variables. Each color of light was the same intensity. Again, the control variable. Describe how the student could make accurate measurements to obtain valid results for the rate of photosynthesis. In order to measure the rate of photosynthesis, you need to measure the volume of oxygen produced for a given amount of time. So how would you do this? So let me draw a diagram to show you how this can be done. There are two typical methods. One is using a measuring cylinder and the other one is using a gas syringe. So let me draw this practical setup. So here's the practical setup that you would typically use. In the practical, you would have the light source and you would place the setup. So there's two different setups. The first one, you've got a beaker with a known volume of water, and you have the pondweed, and on top of the pondweed is a glass funnel, and on top of the glass funnel is a measuring cylinder. So as photosynthesis takes place, you would have oxygen being collected on top of the measuring cylinder. So you can measure the volume of oxygen that is being collected for, let's say, for example, a certain amount of time, like 5 minutes or 10 minutes. Or you could actually measure the volume of gas that is actually given using a gas syringe. I think the gas syringe is actually a little bit more fiddly than using the measuring cylinder, but I think this is actually much more accurate. Now, you would keep the distance between the light source and the setup actually constant, and you would change just the color of the light because you want to keep the distance between the light source and the pondweed actually be the same. So let's go back and have a look at how we can answer this question. Describe how the student should make accurate measurements to obtain valid results for the rate of photosynthesis. So let me write the answer and show you how it can be written. So typically in a question like this, I would actually encourage my students to draw the setup so that it's much easier to actually make reference to. So here's a typical answer. If I'm actually drawing this diagram, I would start off the answer by setup as shown in the diagram. So I'm clearly making reference to the diagram, and allow pondweed to equilibrate to light source. This is very important because it takes time for the pondweed to respond to external stimulus such as a change in temperature or the light. So you would want to give it about a couple of minutes for it to actually get used to the external stimulus. Then collects the oxygen produced using a gas syringe for a given period of time, and I'm giving something like as example 5 minutes, and I'm mentioning the timer. So you don't really have to mention the timer. Writing down 5 minutes indicates that you are actually going to use a timer. So, but I would actually just mention it just in case. Point 3, the rate of the photosynthesis would be equal to the volume of oxygen produced divided by the time taken. The last point is to repeat and calculate average values. So you can calculate average value, do it for each source of light 3 times and calculate the average values, and then repeat it for the different colors of each color of light. That should give you the 4 marks. A student investigated the effect of different wavelengths of light on the rate of photosynthesis. The wavelength of light determines the color of the light. So think back to physics, because you're combined science, you should be able to link to what you're actually reading here to the physics. So the wavelength of light determines the color of the light. Figure 8 shows the student's results. Figure 9 shows the scientist's results. So the first is a bar graph where you have the rate of photosynthesis. You've got red, blue and green. So you can clearly see with blue light, the rate of photosynthesis is the highest, which is about 9. And for red, it's about 7.6. And for green, it's 0.6. So let's have a look at the scientist results. Why are the results for the two investigations presented differently? So the question is asking, why is the student's results presented in a bar graph whereas the scientist's results are presented in a line graph? So first point, students' results are categoric or discrete values, which is the color of light. Those are discrete values. You either have red, blue and green, so you can put it only into categories. Whereas scientists' values of light are continuous values, which is basically numbers. So remember, the independent variable always goes in the x-axis. So the scientist is using different wavelengths of light, and this because both of these are numbers, these are what you call continuous data. Question 4.5. Suggest the range in wavelength of green light. So use figure 8 and figure 9. So you need to look at both of them. Now, as you can see, the rate of photosynthesis is higher at this point. Now, if you compare that to the bar graph, the blue color was the highest. So this would be towards the blue color, and this is the red in between is the green color. So suggest the range in wavelength of green light. So the green light was when the rate of photosynthesis was about 0.6. So if you were to measure it here, the rate of photosynthesis, it's very hard to measure 0.6. That's basically between these points, when you can actually see, right, where the rate of photosynthesis is falling. I would actually take it from there, right, where it's actually dropping. So about, that's 450, 475, so that's about 480, and to the point somewhere here, right, where after that it's starting to increase. So I would say 625. So the range that I would write is between 480 to 625 nanometers. So let's have a look at the marking scheme answer. So on the marking scheme, question 4.1, the purpose of light is to transfer energy of photosynthesis and endothermic reaction. I've combined both of them as part of the answer. And the equation for photosynthesis is carbon dioxide plus water gives you glucose plus oxygen. 4.3, it is to do with how you would measure the rate of photosynthesis. Allow the conveyor to equilibrate in the light. So that's the first point that they have actually said. So, or allow the conveyor in the light for a few minutes before taking measurements. Right? Ignore same light intensity because they've already mentioned that, or anything to do with the distance of light, control temperature, any of those will be ignored, because all of that is nothing to do with measuring the rate of photosynthesis, although I mentioned it as, or identified them as, the different types of variables. The answer needs to be focused on how do you measure the rate of photosynthesis. So first of all, we are going to allow it to equilibrate to the light. Use a gas syringe, right, or you could use a measuring cylinder. Measure the time taken, right, you don't have to measure timer, right. Gas is collected using a timer. repeat the measurements and calculate a mean. So the first point is actually to indicate that you're going to repeat and to calculate the mean, or then you could say that you would discard any anomalies. By calculating the mean, you're actually reducing that effect. Four point 4. The independent variable in the student's investigation is categoric or discrete. So I basically said the student's results are categoric, discrete values, and I refer to the color of the line. And the independent variable in the scientist's investigation is continuous. And I've said whereas the scientist's values of light are continuous values. And 4.5, they have given a range anything between 492 to 577. So they are allowing anything between, so the way I wrote down 480, which is just above that, and this value of, I wrote it down as 625. So anything between these two ranges is acceptable answers. Right, that brings us to the end of question 4 and to the end of this video. Hopefully you found it useful. If you like the useful, please subscribe. Wow, that's the first time I'm actually saying it out loud. So subscribe to my videos, comment on the videos. If you need more help, let me know which past paper questions you want me to go through. I'll gladly go through them. If you want the notes that I'm actually making, I would gladly help you. Email you PDF versions of the exam paper questions. The whole idea of the video is to help you to have past paper questions, have notes, meaningful notes to help you to revise and hopefully to get you better grades. So I'll see you in the third video for this paper for the biology, and that will be the third part of the video, which I'm going to go through question 5 and question 6. Thank you for watching. See you soon.